everyone, Dr. Pinder here again at the Siuslaw Model Forest in Acra, New York. Instead of looking up at the trees today, we are actually going to be looking down at the macro-invertebrates in this stream. Macro-invertebrates are the little creepy crawly bugs that are living in the bottom of the stream, and they are what we call bioindicators. A bioindicator is something that can tell you a lot about the health of the stream, in this particular case water quality, just by looking at the species composition and the general community that you find there. So what we're going to be doing is we're going to be doing some kick netting, where we're going to be sampling some of these macroinvertebrates and seeing what we find to see what it says about this stream's water quality. Alright guys, when you get to your stream site, the first thing I want you to do is describe your site. As usual, I want to know where you went to observe your stream, and I also want a nice detailed description about your stream. So for example, is it really close to people? Does it have a very closed canopy, meaning that there's a lot of leaves covering it, or is it totally open in a suburban park? I also want you to know how much of the water is open versus rock. You can see this is a classic nice Catskill stream where we have a lot of our shale that's popping up here, and we have very little of it is actually exposed water. A lot of it is just going to be running under this creek bed. You would want to note the slope, so if your stream is really steep, is your stream really shallow, and whether or not there's erosion. You can see this bank here has quite a bit of erosion coming out from the tree. You're going to also want to look for what's called as coarse woody debris. That would be any kind of things like wood, logs, twigs, anything that's in the stream itself. That's really good habitat for our macroinvertebrates, and you should note that in your stream description. If we were going to be kick netting this stream to actually quantify the number of species that we would find, we would want to divide our time between all the different types of habitats that were here. So we would want to do an equal number in the riffles area, where we have water flowing over rocks, kind of tumbling like a riffle, or the runs, where we have a more straight shot of water, and the pools, where you have the deeper water, where sometimes you have some more fish. For this, what we're going to do is we're going to try to get our kick net downstream. We're going to work downstream to upstream, and we're going to try to get it so that we, as we kick upstream, whatever's attached to the rocks or under the rocks is going to swim downstream into our net. The best way to do it is to really anchor it at the bottom, and then you're just going to kick away and see what comes out. So now what you do is you just turn out your net and see what kind of stuff you find. And we're going to collect it, and we're going to put it in a white pan so that we can count it and look at it later. Our first sample we got here, we're just going to pick through. What you want to do is have your white pan nice and level, and then you're just going to pick your little critters out. Some of them you can actually get out with your fingers. The goal is not to hurt them. This is a live sampling. We're going to put them right back in the water when we're done. Invertebrate. Just go pop them in the water, please.
And that in the water thing?
Mm-hmm.
Go in. There might not be.
It's crayfish.
There was a fish in there.
We did find some minnows in our kick net, and these guys are black-nosed dace. They're very common stream fish around here, and you can tell that they're black-nosed dace because they have this nice kind of mustache looking snout where they have a little black line on it, and they also have this nice black line that runs down the length of the fish, and they have these peach colored pectoral fins that will turn a really bright color in the summer months during breeding season. So if we were in lab, what we would do is we would spread out along a stream and we would all be kick netting, and we would collect what we would find on the campus stream, and then we would collect them and look at them in the lab. This method though is something that we can also do stream side, so we can look and see what we found. What we found today isn't a huge haul in terms of what we've got, but it is a pretty interesting find. So what I'd like to do is tell you about each one. A couple things you guys will see, we found quite a bit of crayfish, these guys here. We found 5 of those. We did find this one stonefly larvae that is pretty large. We found a dragonfly larva. We found water pennies. And we found, we'll do this guy first. This is a mayfly larva. I don't know if you guys can see, but he has gills that are beating underneath there. And then these guys are caddisfly larvae, these 3. And so this one here is considered, he's kind of naked right now. He's looking for his house. These guys are the actual houses of these caddisfly. And it's really hard to see on the video, but they've actually made little houses using rocks. And you can actually tell the species of caddisfly based on their casing. And so they actually make these really architecturally sound structures out of little tiny pebbles and sticks and leaves. And each species is going to have a different shape of casing depending on the species. Here is a water penny. You can actually see him or her walking on this rock. And I don't know how you can tell from the video, but it's almost completely flush with the rock. So it's well adapted for life in fast-moving water. So one thing you may have noticed is we found quite a bit of diversity in a short time that we were doing our kick netting. The ones that I was asking you to look at for the gills was the mayfly larva. If you were to be able to see it up close, particularly if we were able to go into the lab and look at it under the dissecting scope, you'd actually see these little parts here beating like crazy, and those are the gills. And so what they're doing is by moving that, they're passing water over the gills really quickly, and then what they can do is get more oxygen out of the water. So just like we get oxygen out of the air, these guys are going to be getting the oxygen directly from the water. And so the sensitive species that we are looking for, the ones that are generally considered species that you'll find in good quality streams where we have a lot of oxygen, that's the ones that are going to be taking the oxygen right out of the water. There's 3 particular groups that are really important. The mayflies, which are the Ephemeroptera, the stoneflies, which are the Plecopteras, and then the caddisfly, which is Trichoptera. Sometimes you may see something called EPT richness, and that's for that Ephemeroptera, Plecoptera, and Trichoptera richness. So how many species of each of these groups did you find? Because they're a particularly sensitive group, because they are trying to get that oxygen right out of the water. Something like a diving beetle or a mosquito, those larvae are going to be able to suck air or get their oxygen right out of the air, just like we would. Little mosquito larvae, when they're hanging at the surface of the water, what they're doing is they're using a snorkel to get atmospheric oxygen rather than water oxygen. So these guys are a little bit more sensitive, because if the water level of oxygen is too low, then they can't get the oxygen they need to survive. So that's why generally you'll see mosquitoes in anything from like tire rims or really stagnant water. They don't need a lot of oxygen, and that's a good species to think of that you don't think of as something in a really high quality water. Yeah, they could live here, but they are going to be found in really trashy places as well. Whereas these guys are not going to be found in the rim of a tire ever, just because they can't get the oxygen that they need. Alright guys, that does it for our time here at the stream. I'm going to take our results back and tally them up, and I'll share them with you on lab for Thursday.